Good morning, children. Today we are going to do our first practical from the O labs. The first practical that we are going to do is study of pollen germination. Now, this is our objective or aim of the experiment what that we write. Our aim is to study the pollen germination on a slide. Then children, this is the theory. You have to do go through the theory properly because your vivas can be asked from these theory, these portions of theory. So pollination is a very important part of the life cycle of flowering plant. We know that. Pollination is the transference of pollen grain from the anthers of a flower to the stigma. You know what is pollination. We have done different types of pollination also. Transferring of pollen grains from the stigma, from the anthers to the stigma of the flower is called pollination. Flower mediated by abiotic or biotic means. Now the abiotic means we know like wind, water are the abiotic means, and any living organism, if that living organism is helping in the transferring of pollen grains, that is the biotic means, like insects, birds, animals, mammals, right? So abiotic means the pollen is carried by organism but through means such as wind and water, that is abiotic, not carried by humans but by wind and water, and those which are carried by animals, insects, birds, that is biotic. So we all know this, we have done this. So in flowering plants, however, the ovules are contained within a hollow organ called pistil, and the pollen is deposited on the pistil. Now you have to remember that pistil has a surface which is receptive, which is ready to receive the pollen. So on the stigma, the germination, many pollen grains will come and land on the stigma, but very few can germinate on the stigma. So the germination of pollen grain begins, and how does it begins? What are the requirements? By the absorption of water and nutrients. So for germination of pollen grains also, water and nutrients are required. This is a very important topic. Okay, and the pollen grain produces a tiny pollen tube, and this pollen tube will start its journey from the stigma, and it will pass on through the style and reach the ovary. We all know this. So the tube cells enlarge and comes out of the pollen grain through one of the germ pores to form the pollen tube. So you know what is a germ pore. We have done this when we was doing the structure of pollen grain. So there is a hole or there is a pore called germ pore through which the pollen tube will come out. So this is the germ pore, and the pollen tube has started its journey. You can say it has started to grow, and it will reach the ovule. Okay, so we also know that making the possible the development of embryo and other cells combined. Now here we have also done the structure of pollen grain, and we know that it is divided into two cells. One is the generative cell and the other one is the vegetative cell. We have done this in theory. Now the generative cell also passes into it. It soon divides into two male gametes, we know that, and in the act of double fertilization, one of the sperm cells within the pollen tube will fuse the egg. So two male gametes are traveling inside the pollen tube. Two male gametes are traveling inside the pollen tube. Out of these two male gamete, one will fuse with the egg cell of the ovule, and the zygote will be formed, and that will develop into an embryo, right? And the other cell will go and fuse with the polar nuclei, two polar nuclei. So already two polar nuclei were present in the nucellus of the female part, we know that, and now the third nuclei, that is the third pollen grain, has also reached. Now it has been converted into a triploid pen, that is primary endosperm nucleus. So this all has been done in your theory, right? And the pen is surrounded by PEC. What is PEC? Primary endosperm cell, and that is actually the endosperm where the reserve food is being kept for the developing zygote and the embryo. The growing ovule then transfers into a seed, and we can say, and the ovary will get transferred into an fruit, right? So these are the theory of study of pollen germination. Now let's see the procedure here, children. If you see the procedure, let me show you. So fresh mature flower, 10 milligram of boric acid, distilled water, glass slide, 30 milligram of magnesium sulfate, 1 glass rod, 20 milligram of potassium nitrate, a dropper, 10 gram. Now here it is gram, other were milligrams. You see, this is gram of sucrose and a compound microscope. Now, what you have to do is prepare the pollen germination medium by dissolving 10 gram of sucrose, 10 milligram of boric acid, 30 milligram of magnesium sulfate, and 20 milligrams of potassium nitrate in 100 ml of distilled water. So what you have to do, you have to dissolve all these contents into 100 ml of distilled water so that your nutrient medium is ready. Now this, what are you making? Actually, you are making the nutrient medium. Now using a glass rod, stir the solution to mix it well, and using a dropper, take some of the nutrient solution, the solution that you have made just now, take few drops of it and put the 2 drops on a clean glass slide. Now take a mature flower and dust the few pollen grains from its stamen on the drop of the glass slide. The drops that you have taken, the nutrient medium के जो drops आपने लिए, उसी nutrient medium के drops के ऊपर थोड़े से pollen grains आपको dust करने हैं, right? And after 5 minutes, place a glass slide on the stage of the compound microscope, and you will observe through the microscope after a half an hour. Just keep it for half an hour and observe after that. Okay. Now here stimulator procedure as performed on online is these are the certain you can say they will steps which you have to keep in mind. Click and drag the dropper towards the beaker containing germination medium to take the medium. Drag the dropper towards the glass slide to pour a few drops of germination medium on the glass slide. Okay, drag the flower towards the glass slide to dust the pollen grain. Then drag the place. These are the procedures. Same thing, but they have written it here through an online labs. They have used it. Now, what are the observation after half an hour? What will you observe when the whole procedure has been done? What is the observation? The pollen grain is uninucleate. It has only one nucleus in the beginning, and at the time of liberation it becomes 2 cells, है ना, दो हो जाते हैं. One is the generative and the other one is the vegetative. So in the nutrient medium, pollen grain germinates. The tube cell enlarges and comes out of the pollen grain through a germ pore to form a pollen tube, and the tube nucleus descends in to the tip of the pollen tube. Generative cell also passes it into that pollen tube. Soon it will divide into two